Okay, welcome back to the final video in this module where we will finish our discussion of antigen receptors by looking at molecular signal transduction downstream of the TCR and the BCR. So we're going to start by looking at the BCR, which remember is a form of immunoglobulin, and this is something expressed on the surface of B cells, which allows them to interact with their antigen, recognize their antigen. We looked at what antibodies do already in a previous video when they bind to antigen, the types of effector mechanisms that they mediate. But let's think now what happens downstream of BCR activation when the BCR binds to its antigen. We saw in the last video that the T cell receptor complex had a variety of other molecules that were important for its ability to transmit an intracellular signal. The same is true for the BCR, although it's a little bit simpler overall. So the BCR itself is primarily made up of the immunoglobulin molecule, which is extracellular and really just serves to be able to recognize antigen. In order to transmit an intracellular signal, the BCR relies on two additional molecules, which you could sort of think of again as co-receptors. They are referred to as immunoglobulin alpha and immunoglobulin beta, maybe just Ig alpha and Ig beta. Again, not IgA, which is a class of immunoglobulin. So it's important here that we say Ig alpha and Ig beta, which are these co-receptors for the BCR complex. What's key about these two molecules is that they again have these deeper cytoplasmic tails, which allow them to interact with signaling molecules in the cytoplasm of the B cell. And the way that they facilitate those interactions are through particular sort of portions of these tails, which we refer to as ITAMs. ITAM stands for immunoreceptor tyrosine-based activation motifs. So these are tyrosine residues within these tails which serve as platforms essentially for phosphorylation. And when the ITAMs are phosphorylated, that is going to facilitate the recruitment of other signaling molecules, which will continue the transduction of a signaling pathway, which we'll see sort of play out here in a minute. What I want to point out about B cells in particular is that in order for the BCR complex to fully signal, it also requires crosslinking. We saw an example of this with Fc receptors. They require crosslinking to transmit intracellular signaling. B cells are the same. And the way that happens is that, remember that immunoglobulins, BCRs, are multivalent. And they can bind to polyvalent antigens. And when two BCRs bind to the same antigen, that again is going to pull them closer together in physical space. And when they become physically opposed to each other, that's going to bring the ITAMs on their intracellular chains close together. And when these molecules congregate in the plasma membrane, that facilitates the activity of a particular Src family kinase, which is sort of embedded in the plasma membrane, known as Lyn. Src family kinases are associated with the activation of a number of different types of receptors in the immune system, and probably most famously B cells here. And Lyn is a particular Src family kinase that is probably the most important one that initiates signal transduction downstream of the cross-linking of B cell receptors. There are some others I'm not mentioning here, but I think Lyn is the most important one to note. A major substrate for Lyn, I should say that Lyn is a tyrosine kinase, and so it can phosphorylate these tyrosine residues, these ITAMs, on the cytoplasmic tails of Ig alpha and Ig beta. And you can see these phosphate groups being added here in this image. So now these phosphorylated ITAMs can serve as sort of docking platforms for additional signaling molecules to bind. But before we move further downstream, now let's look at the the T cell receptor complex following the B cell receptor complex following its cross-linking, and some of the additional molecules that then get recruited. So I mentioned that phosphorylated ITAMs serve as a kind of scaffold for additional things to start to bind. One of the major things that is able to bind to these phosphorylated ITAMs is now another kinase, which belongs to a family of kinases known as Syk. So a Syk or SYK family kinase. In B cells, the Syk family kinase is actually Syk itself. The kinase that kind of gives the family its name. But what I want to stress here is that Syk family kinases, including Syk, have two binding domains as part of their structure known as SH2 domains. And these SH2 domains facilitate binding to phosphorylated ITAMs. So Syk is inactive in its normal conformation. So when it's not bound to anything, it does not have any kinase activity. It's inactive. But when the SH2 domains bind to the phosphorylated tyrosine residues of ITAMs, that induces a conformational change, which sort of unlocks the kinase activity of Syk and allows it to go on to phosphorylate downstream targets. So you can see here that this cross-linked BCR, now its ITAMs have been phosphorylated upstream by Lyn. These phosphorylated ITAMs can now recruit Syk, leading to its activation. And Syk has many substrates, many things that it can phosphorylate. Again, for this introductory video, I think the most important thing that we can point out that it activates next is an adapter molecule. In B cells, this adapter molecule is known as BLNK, or B-cell linker protein. And this phosphorylated adapter molecule, this linker protein, then goes on to recruit a bunch of other downstream signaling molecules, which facilitate all the things that need to happen intracellularly within the B cell to render it activated. We'll come back to that point at the end of the video. But from here, I just want to say that you'll see a similar process play out in T cells, where we activate a Syk family kinase through its association with phosphorylated ITAMs, and then that Syk family kinase is going to activate an adapter molecule in B cells that is known as BLNK, and then the adapter facilitates further downstream signaling. So now that we've walked through that in the context of a B cell, let's look at similar processes as they play out in T cells. So again, we're going to use the CD8 T cell as our exemplar here, but we could draw a very similar cartoon with a CD4 T cell. The only difference would be you would have CD4 here instead of CD8, and you would have MHC class 2 instead of MHC class 1. But all the intracellular stuff is identical. So just like we saw with Ig alpha and Ig beta, the cytoplasmic tails of the CD3 co-receptor complex also have ITAMs, in fact, many ITAMs. The zeta chains, which remember sort of extend deeper into the cytoplasm, especially have many ITAMs. And these ITAMs are going to again serve as a scaffold for phosphorylation of tyrosine residues, and that is going to allow the recruitment of downstream signaling molecules. In T cells, we have a different Src family kinase that is going to phosphorylate the ITAMs. In T cells, the most important Src family kinase is known as Lck, or LCK. And whereas in B cells, the Src family kinase was Lyn, Lyn, Lyn was sort of more anchored in the plasma membrane and sort of floats around by, sort of through the plasma membrane, sort of a little more autonomously. The Src family kinase in T cell is actually physically associated with the co-receptor. So you can see here that Lck is physically associated with CD8. And I should say in CD4 T cells, you find Lck associated with CD4. So CD8 and CD4 are interchangeable here. But Lck is going to be our Src family kinase, and it is going to phosphorylate the ITAMs on all the various cytoplasmic chains of CD3. And you can notice now we have all these phosphorylated ITAMs, and that's going to allow us to recruit further signaling molecules and transduce this signal further. So the way that's going to look is we're going to have another Syk family kinase. The Syk family kinase that's most important in T cells now is not Syk, but rather a Syk family kinase known as ZAP70, ZAP70. And as a Syk family kinase, it also has SH2 domains. And when these SH2 domains are not bound to phosphorylated tyrosine residues, the overall kinase molecule is inactive. So before it binds to the ITAM, ZAP70 cannot phosphorylate anything. But when its SH2 domains come in contact with the phosphorylated tyrosine residues of the ITAM, that facilitates a conformational change, which activates the kinase activity of the enzyme. And again, ZAP70 also has many downstream substrates that it can phosphorylate. The most important of which, I would argue, in T cells is an adapter molecule called LAT. LAT stands for linker for activation of T cells. So just like in B cells, we see that downstream of our Sick family kinase, there's an adapter molecule, and that adapter molecule is going to recruit a bunch of molecules then that facilitate further downstream signaling. So if we take those general principles from both B cells and T cells, and we try to schematize the flow of signaling that happened in both cases, we get something that looks like this. If we take a table here and we put B cells and T cells into their own columns, we see that in order to activate either type of lymphocyte, either B cells or T cells, we need to start with an antigen recognition event. That's going to involve the binding of antigen, some kind of soluble antigen usually, to the B cell receptor and inducing its crosslinking, or some type of presented antigen onto the T cell receptor, and that is going to be facilitated by MHC, either one or two. Either way, the TCR, the BCR, the cells that are expressing those receptors need to see their antigen in order to become activated. When we have a successful antigen recognition event, the most important thing that happens next is the activation of a Src family kinase. In B cells, that Src family kinase is Lyn, and in T cells, that Src family kinase is Lck. The major differences here is that Lyn is tethered into the membrane, but typically is sort of diffusing around, floating around in the membrane on its own terms until it comes into contact with or comes into close proximity with cross-linked B cell receptors, whereas Lck is very similar in terms of its function to Lyn, but within T cells, Lck is physically associated with the co-receptors CD4 and CD8. In either case, the activation of the Src family kinase is going to phosphorylate ITAMs associated with the various co-receptors that we find on both the BCR and the TCR. So in the B cell, the ITAMs are found on the cytoplasmic tails of Ig alpha and Ig beta, and they are going to be phosphorylated by Lyn. And within the T cell, it's going to be all of those cytoplasmic chains associated with CD3. So remember, there was an epsilon delta heterodimer, a gamma epsilon heterodimer, and then two sort of larger zeta chain homodimers, and all of which have ITAMs, and which are going to be phosphorylated in T cells by Lck. In both cases, these phosphorylated ITAMs facilitate the recruitment and activation of a Sick family kinase. In B cells, that is Sick itself, the kinase for which this family gets its name, whereas in T cells, the Sick family kinase is going to be ZAP70. And the activation of either of these Sick family kinases, again, is going to result in the phosphorylation of many downstream targets, but probably the most important of all is going to be the phosphorylation of key adapter proteins within these cells. In B cells, that's going to be BLNK, B cell linker protein. In T cells, it's going to be LAT, linker for activation of T cells. And these adapter proteins are then going to facilitate the recruitment of many other signaling molecules downstream that facilitate the activation of the cell. And so for the purposes of this video, I'm going to not really go into all the dozens of downstream molecules that we could sort of fill in the blanks with downstream of BLNK and LAT. But I just want to make the point that both BLNK and LAT, again, these molecules are uniquely found either in B cells or in T cells, but they have overlapping functions. What they do within those cells is, again, as adapter molecules, they serve as sort of a physical platform that a bunch of downstream enzymes and other signaling molecules can bind to. And this sort of, it serves as a platform through which a lot of downstream signaling can then occur. And we refer to the large kind of quaternary complex of multiple enzymes that all come together physically connected to these adapter molecules as signalosomes. And these signalosomes, you can think of there being a B cell signalosome and a T cell signalosome that are nucleated or initiated by either BLNK or LAT, respectively, for B cells and T cells. Again, the purposes of this video is not really to talk about what all of these new molecules are, but a conceptual point that I want to make is that the formation of the signalosome downstream of the antigen receptor is going to unlock what we could think of as 4 modules of effector function, or 4 modules of downstream signaling that are going to mediate the various things that the B cell or the T cell needs to do once it sees its antigen. Those 4 modules of signaling include things like metabolic reprogramming. This typically involves the upregulation of cellular metabolism. This is going to be important because the cell, once it sees its antigen, it's going to need to start making a bunch of new things. Usually it's also going to start dividing and proliferating, and that's all going to take a lot of ATP and other kind of metabolic intermediates to provide the building blocks for all these new molecules that need to be synthesized. And so the signalosome is going to have one major result, which is the enhancement typically of B cell or T cell metabolism. Another major outcome is going to be transcriptional activation. By this point, that might not be surprising. We see that a major output of almost all immunological receptors is transcriptional activation. And there's a similar concept here. Typically, the B cell or the T cell needs to start making new types of molecules, new types of cytokines, or cytotoxic effector molecules, and that's going to require de novo transcription so that those molecules can be translated and produced. So there are going to be many transcription factors found downstream of the signalosome that are going to facilitate a new transcriptional program associated with lymphocyte activation. An additional module we could think of as cytoskeletal reorganization, and cytoskeletal reorganization serves multiple purposes. One, as we've seen in other videos, cytoskeletal reorganization is important for cellular movement, and typically a B cell or a T cell, when it sees its antigen, it then needs to move around. If it were naive when it became activated, it usually needs to leave the lymph node or leave the secondary lymphoid organ where it is so that it can migrate to the tissue where the infection is, so that it can actually do whatever it needs to do at the actual site of infection. But beyond that, these cells also need to do things like perform diapedesis, sort of squeeze between the walls of blood vessels so they can access tissues. That is also a process that needs a lot of cytoskeletal rearrangement in order for T cells, or especially for T cells, to sort of do that squeezing process. Cytoskeletal reorganization is also important for things like endocytosis, which B cells tend to do when they recognize their antigen that activates their BCR, but then they also endocytose that antigen bound to the BCR so that they can then serve as professional antigen-presenting cells. And we'll see how that process plays out in a future video. Finally, a major module of function that is unlocked by the B cell or the T cell signalosome is the activation and expression of molecules called integrins. Integrins we haven't really talked about yet, but the main point that I want to make is that these are molecules that help lymphocytes sort of stick to things and sort of tether themselves physically in space, either to cells that they want to continue to interact with or to tissue microenvironments, the extracellular matrix, where they want to hang out. And so integrins, for example, are important for sort of holding on to antigen-presenting cells long enough for antigen presentation to occur. And so integrins can serve that function on a kind of cell-to-cell basis, but they're also important for things like staying in the correct area of the lymph node or moving along the walls of blood vessels, despite the fact that blood is rushing past at a very high rate. There's a lot of shear force within the walls of blood vessels, and integrins can sort of help lymphocytes sort of hang on and stay in a location where they might want to stay for some reason in terms of their development or their effector function. And so this is a final module of activation, again, that is unlocked by the activities of the B cell or the T cell signalosome. Okay, summarizing this final video in this module, we started with BCRs and we saw that they require cross-linking in order to initiate downstream signaling. And when they cross-link, a kinase known as Lyn its activity is facilitated, and it can then phosphorylate tyrosine residues known as ITAMs on the co-receptors Ig alpha and Ig beta. And these phosphorylated ITAMs recruit a Syk family kinase, in this case known as Syk. We saw that antigen recognition by the TCR, even though it looks a lot different on the extracellular side, there's very similar things happening intracellularly. We have a Sarc family kinase known as Lck in this case, which will phosphorylate ITAMs as well. Within the T cell, those are going to be the chains of CD3. And those phosphorylated ITAMs on CD3 then recruit a different Sic family kinase known as ZAP70. Both Sic and ZAP70 have a similar function in that they phosphorylate multiple downstream substrates, the most important of which I would argue are the adapter molecules, BLNK, which stands for B cell linker protein, as well as LAT, which stands for linker for activation of T cells. And these adapter molecules nucleate the formation of signalosomes, which recruit many downstream signaling molecules, which unlock four kind of modules of intracellular signaling, which are important for mediating lymphocyte effector function. Some examples of those were cytoskeletal reorganization and integrin expression. These were important for things like cellular movement and kind of interaction with the cellular environment, as well as metabolic upregulation and transcription factor activation. And these are going to be more related to the processes of proliferation and de novo synthesis of cytokines and other antimicrobial effector molecules. Okay, that covers it for our discussion of antigen binding and antigen receptor signaling. I'll see you in the next module.